All right, hello! Today's adventure on World Water Monitoring Day is to monitor some water. So let's learn how to do that. One, what does it take to measure water? What kind of things can you measure about water? And what is really interesting to know? So, if you take a look at this riverway, this is part of the Jordan River, you might notice several things. For example, what plants are around the banks? How fast is the water moving? What animals are flying or floating? What kind of signs of humans do you see? Today, we're going to zoom in to the actual inside of the river. So we're going to really look down. We're going to take some water samples and we're going to measure things like temperature, dissolved oxygen. Can you think of why oxygen is important to measure? All the aquatic life that lives in the river needs oxygen, just like we do. We're also going to measure things like nitrogen and phosphorus, which can be indicators of how much, here's some bees. The nitrogen and phosphorus is an indicator of how much nutrients are in the water. So if there's lots of nutrients, oftentimes we'll see a lot more algae growing. And the nitrogen and phosphorus can come from places like runoff of farm fields. We're also going to measure the turbidity, which is how well you can see through the water. All right, are you ready to learn how to do all of this? First, go ahead and make a scientific question. So that's going to look something like, what is the effect of blank on blank? What is the relationship between two different variables? Or what factors cause? So I'll give you 3 examples. Okay, the first example, what factors cause? We're going to go backwards. What factors cause the stream to be considered healthy or not? That's maybe a research question. Go and I suggest you listen to Elise Hinman from the Department of Water Quality talk about how we determine if streams are healthy. She does water monitoring all over Salt Lake, and she can tell you a little bit more about what standards exist for water quality. You might also want to know, what is the effect of something you observe? What is the effect of the type of waterway? So for a type of waterway, this one is a river, and if I run across here, you can see some native plants that are being restored. You could wonder about that too. What's the effect of having native vegetation around a body of water? So here's getting back to the original question. What's the effect of the type of body of water? So this is a wetland. You can tell it looks vastly different from the river. So what's the effect of the type of body of water on, and then all your different variables? So dissolved oxygen, nitrogen, phosphorus, temperature. What do you think? What's your hypothesis? And then I might want to know different variables and their relationship between them. So what's the relationship between temperature and dissolved oxygen? There are a couple things to know about that, and let's go explore. Why might we care about all these different factors? Well, if you want your stream to be healthy, or your wetland to be healthy, you're going to want to know that there's enough oxygen for animals to live. You're going to want to know if there's too much nitrogen, and so then there's algae blooms, which end up causing dead zones, because the algae, when it decomposes, takes up a lot of oxygen, meaning that other things can't live. So we're going to learn about this and so much more, but let's dive into the actual tools that we use to measure, and we'll do a water quality sampling together. Okay, so step 1, gather your water. So you're going to take some sort of vessel, and if you don't have a fancy one like this, you can just use a water bottle or something like that. Ooh, notice a little water strider. Hey, buddy. How do you think that they can stand on the water like that? What do you know about water striders? Check out that lesson in the water module. Okay, so make sure different safety things. Make sure you're not going to fall in, that you're stable on the surface, and try not to disturb any of the other factors, the sides of the banks. Don't cause erosion to start happening there. Okay, so I got my sample of water. Now let's go measure it. First, I want to make some observations. What do you notice? Just like right off the bat. What's true about this water that you see compared to other water? I maybe don't want to drink it. Well, that's a start. That's kind of an inference, right? We're adding our own knowledge onto this idea of something that we observed. So we might all agree that this isn't 100% clear. But to say that you should or shouldn't drink it, you might need some more evidence for us all to agree on that. Okay, so I'm going to take my second sample over here across the pond, and I'm going to write down any observations that I notice. For example, it seems like this is covered in what? Some green substance. If I look closer, I think this is called pondweed. Okay, so I took my sample. Here it is, yum, yum, yum. And let's go compare it to the river. So we're comparing a wetland pond to a river. Here we go. So here's the very efficient lab bench that we have, and we have all of our tools laid out here. So let's walk through them. First, let's just start with temperature. This is maybe one of the easiest things that you can measure if you don't have all these fancy tools. So you might use a thermometer to measure that. But if you do have fancy tools, this is one of my favorite fancy tools, which is a temperature gun. Okay, so this, you just don't shoot it in people's eyes, right? But let's see, I hope mine is, oop, we maybe need more batteries, which is awesome because we have the mercury thermometer instead, which sometimes gives better results in my opinion. Okay, so let's go ahead and measure the temperature. Let's go through our tools first. Temperature, we can measure pH. So one thing we might find, well, let's just leave this up to exploration. There's lots I can tell you about how all these variables interact, but that's the fun of going out and measuring these yourself, so you can see the relationships here. pH, we can go from 0 to 7. This one's from 0 to 14. On the back here it shows 7 to 14. So 0 to 7 is acidic, and 7 to 14 is basic. What do you think is the best pH for animals and fish and macroinvertebrates to be healthy? Then we can measure phosphate and nitrogen, those two elements that compounds. And this is actually pretty fun and pretty science. We'll get into this, but there you go. There's nitrate. Here's the phosphorus kit. Awesome. And the final thing is dissolved oxygen. Okay, so we'll measure some dissolved oxygen. How much is in the water? And the final thing we're going to look at is the turbidity, how much we can actually see through the water. So if we take a look at our sample, and it's in kind of an opaque bottle anyway, but you can see that it's maybe pretty see-through. Like if you were swimming through this and you opened your eyes, would you be able to see very far? That's what this optical refractometer is going to tell us. All right, and our most important piece of equipment is our notebook. So we're going to start by taking notes, and I'll show you how to do this. You want to make sure you know where your sample's coming from. So I'm going to label the sample Wetland Reserve on the Jordan River. And then I want to take some data on the actual physical environment. So I notice that today it is, so make sure you write your date. Today is World Water Monitoring Day, 9/18/2020. My physical environment, it is currently, if you see, it is currently pretty cloudy. Okay, so I want to write down what the weather's doing. You might want to write down if it's rainy or not, or if it's windy. So it's not windy. And you might also describe this as being still, not windy or still. And then you want to get a sense of the temperature. Do you think there's going to be a difference between the air temperature and the water temperature? What do you think it's going to be? But if I start looking at my thermometer, I notice that first I want to know that I'm measuring in, can you see this? Degrees Celsius. Okay, and I look at how far up the mercury has risen, and we're at about 20, you see that? 20 degrees. Okay, so I'm gonna write down 20 degrees C for the air temp. And the time of day, so it's about noon. So I'll do 12 p.m. Okay. All right, now we're ready to start testing the water. All right, so first and easiest thing, going to put my thermometer in there and wait to see if there's any changes, and then measure my temperature. Making sure that the ball of the mercury is down in the water. It's pretty important. So I already see it's dropping quite a bit. We were at 20 degrees for the air temp. And still it's going lower and lower. Let's see here. All right, it hasn't moved in a little bit, so I'm gonna go ahead and write down, if I go 0 to 10, each one of these markings is 1, and then this would be about 6.5, between 6 and 7. Okay? Measuring for my pond sample, and ooh, wow, pretty interesting. Let's see, it's getting cooler. But not quite as cold as the river. Very interesting. So I would say we're at 8 degrees C. So now let's go ahead and measure the pH. So I have my pH indicator strips. I'm going to take one out. And this is what I'm going to put in there, and it's going to change colors based on the pH of the water. Okay, so did you notice that color change? Kind of hard to see on my camera. Okay, so as I put that down, I'm gonna then compare it to my kit, and that's what this key is for. So maybe it's hard to tell with the colors, but I'm gonna make the executive decision that we're actually at a pH of 7, which is neutral, right in the middle. I'm gonna check the back side. Also looks like it agrees. Okay, so I'm gonna go with 7 for the pH. All right, so we dipped our strip in, and we can see the color change from this to that. So now we want to take that and compare it to our pH indicator guide, and you can see that different levels of this would change. So if I line this up, it kind of looks like pH 7. What do you think? I have it upside down, but there we go. Also maybe matches a pH of 7. All right, so I'm going to go ahead and write that down. Here I made just some notes. The water temperature, I got my pH of 7. All right, and now let's go on to phosphate, nitrogen, dissolved oxygen. I'm so excited about these because look how pretty this is. All right, this is all very pretty science. All right, let's dive into how this works. Okay, so with phosphate, read the directions here, but what we're gonna do is we're gonna take this sample cup. Here's our sample cup. And then we're gonna fill it and add 2 drops of the indicator solution. So that is this one All this is hard to do with one hand. That's okay. Ooh, pretty fun. Pretty fun here. Okay, so we can take this out. Actually, let me take a minute. I've never done this before, so let me figure out what's going on. Step one, fill this up. And I'm actually just gonna wash it out from before, so I'm gonna fill it with the water and then wash it out one more time. You'll see this go down. I'm gonna add my indicator activator solution, 2 drops. 1, 2, oh. Okay. I'm gonna cap the sample cup and shake it to mix the contents. Cap it. Here we go, here we go, here we go. This is hard. Wow, this is actually super cool. So what's actually gonna go down here is I'm gonna put this in and then push it up against the side to break the tip, okay? And then watch it fill up with water. So what's happening here is there's a higher pressure on the outside where the water is, and it's going to force it up into the tube. Okay, so now my ampule is filled and I'm going to invert it a couple times, meaning go up and down and allow the chemicals to mix together. Okay, and then from here, let's see, I don't really detect a color change, which is maybe okay. What we're going to measure this against is some solutions that have already been made with different levels of phosphorus to see what is the actual level of phosphorus in our sample. Okay, so that's where I'm going to take, let's see, does this work with the camera? Alright, you can still see this? Yes. And I'm going to put it in here, pop this lid off. Oh nope, I'm just going to put it in. Put it in like that. So the surface tension keeps the water from escaping. Those water molecules like to hold on to themselves better than they hold on to the surface of this glass so that they don't fall out. Okay, so I put it in and now I look to see and sort of see there. It's hard to tell, but you compare then this to the colors around it. Oh yeah, you can actually definitely see a color change. So I'm maybe gonna make the decision, ooh, it's like a different color blue, but it's a little bit darker. So I'm gonna go around and see that darker blue is at 0.8. See if you can see that. This goes to that. Alright, and if this is getting frustrating that you can't see it in person, come out tomorrow if you're in the Salt Lake area, and we'll do this water monitoring together. Okay, so cool. So we figured out the phosphorus is at 0.8. What actually went down here is a really cool chemical reaction. So it uses the stannous chloride chemistry. So an orthophosphate reacts with ammonia molybdate to form molybdophosphorus acid, which is then reduced by stannous chloride to the intensely colored molybdenum blue. And that's the color that we see. So that color then is directly proportional to the phosphate concentration. Okay, so we got a phosphate concentration of 0.8, and that's measured in parts per millimeter. Or milligrams per liter. Parts per million, sorry. Parts per million. So there's 1.8 parts of phosphate in a million parts of water. That's what that means. Or there's 0.8 milligrams per liter of water of phosphate. All right, cool. So let's go ahead and write that down. This sort of means nothing right now until we compare it to some standards. And we'll have to go look up, like, what's an appropriate level of phosphate? But I'm going to make sure not to leave naked numbers. And so 0.8, we just said ppm, parts per million, or milligrams per liter of PO 4. All right, let's go on to nitrate. Okay, so here on the left is from the pond sample, and the right is the river. So let's go ahead and do the pond. We'll do the pond first. We'll measure it up against that. Going each one around and seeing. For me, it looks like this would be about 0.6. I think it matches best with that one. Right around 0.6. Okay, so let's talk about our next test here, which is dissolved oxygen. So there's a delicate balance that goes on with oxygen in the environment. You have animals that breathe oxygen, taking in, release carbon dioxide, and then the plants take in the carbon dioxide and release oxygen. So anything that upsets this delicate balance can be really devastating to the whole ecosystem. So let's take a look at what the dissolved oxygen is like in our 2 different samples. All right, so here we go measuring our dissolved oxygen. Go ahead and put this in and take a close look at how fun this reaction is. And what we'll be able to see here now is how much dissolved oxygen is in the water. It's a little bit tricky sometimes because of how oxygen saturated our environment is, the water becomes saturated. So it's important to try not to disturb your water sample when you grab it. And let any solids sink to the bottom, so that's not affecting it either. But then we can go ahead and measure up against our water samples here, so I think we're pretty high in oxygen. And so lakes that are high in oxygen but low in nutrients are called oligotrophic lakes. Lakes that are high in oxygen or are high in nutrients but fluctuate in the amount of oxygen are called eutrophic. Can you think of which one would support more animal life? A eutrophic or an oligotrophic? A eutrophic lake would, because it is, like we can see out there. Oop, that's better. And then let's go ahead and test it out. So on the right is the pond. You can just kind of see the pond looks a little bit lighter already. So let's go ahead and measure that pond one first. Which one does it seem closest to? I'm gonna put it around here. It's a little lighter than that. Okay, I'm gonna put it at 4. Yeah, it's darker than that one. Okay, so 4 parts per million for the pond. And maybe around 6. Around 6 for the river. Here's our refractometer. So when we look into this plate, we can see a level, and you can, let's see here. So I just put my sample down, and you can see here, let's see, there you go. See that line at the bottom? Sodium chloride. I'm not quite sure why it says sodium chloride, but what this is measuring is the light that gets through. So if there's more things in there, if it's more saturated with other particles, you'll be less likely to be able to see straight through there. So this is measuring the amount of other particles that are in the water. So this says it's pretty clear. Another measure for that would be turbidity. Sample on, close this disc, and look through the hole, up into the light. And you can see this is for the pond. It's also pretty clear. How would this change if we had gotten some of the duckweed in there? Or if this was a dirty, murky water? Alright, just summarizing the biological survey, counting 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 ducks. Mallard ducks. Let's see, 2 males and a lot of females. And some of them look a little bit darker in their tail feathers, the females, than others. Just variation, or could there be something more to that? So I came across this fellow,
Austin.
Austin, and you're doing what?
So I'm a natural resource resources technician, and I work for the city of Salt Lake. And so basically, we're in charge of keeping these open spaces for everyone to enjoy. And my specific job is to make sure we have high biodiversity in areas like these.
What's biodiversity?
So biodiversity is basically when you have different species composition throughout your ecosystem. So you don't have one invasive species or one monoculture or something of that.
So you want lots of different types of organisms.
Lots of organisms, lots of plants, and it keeps things healthy and resilient to climate change and things of that nature.
Oh, cool.
Yes.
So you were just telling me you wish you could do more of the science that we're doing here.
Yes, very much so.
And what would you be looking at, or what kind of science would you want to be doing?
So I went to college and I studied water quality measurements and making sure we have good, clean water for plants, animals, and people. And soil quality, so looking at soil health, making sure we're looking at things like compaction and microbiome that's within the plants.
Oh,
yeah.
That's so cool.
Yeah.
So what's your assessment on this area? We're about to analyze our data, but we're looking at the difference between this stream over there and the pond over here.
So I would say the first thing that you notice is you have a lot more aquatic organisms there. We actually have an overgrowth of duckweed. It's a perfectly fine plant, it's not invasive, it's native to the area, but when you don't have high water flows and you get hot temperatures like we have in Utah, you're going to get an abundance of growth that you can see there. So we're trying to get a hold on that without using any sort of chemicals, because we have treated it in the past. But
what would you do?
So we have treated it with fluoridone, which specifically targets duckweed and Eurasian water milfoil. So
Whoa.
It's those. But it's important to control your aquatic plants as well, because I mean, if you look at that, it's completely strangled and you're not getting your sunlight, which I mean leads to a monoculture. That's all entirely one plant essentially at this point. So,
oh, awesome. Well, thanks so much for teaching us more, giving some depth to this experiment. Do you have any advice for youth, people watching this?
I would say if you have a strong passion for the sciences, that is something you should definitely pursue. Because this area actually, believe it or not, used to be like a railroad, essentially. And
really?
So the city bought it, and we're trying to restore it back to a native little riparian area with a wetland involved.
Oh, that's awesome. Well, it's so fun to be out here, and it's beautiful. So, thanks for doing the work you do.
Yeah, of course, no problem.
Okay, so then we ran into another person who has something to say about this here experiment.
Oh, cool. So I was just curious.
Yeah.
We're collecting water from the Jordan River in this pond at the Fife Preserve, and I'm curious to see what the results are, because this water, the Fife Preserve's lake, the stream has a big oxbow right here, comes around all the way around this way, and the water that's in this lake is coming from the Jordan River that loops around this way. So I was just curious to see what the different results are.
And what's your hypothesis?
I'm assuming there's going to be a lot of nitrogen and phosphorus in here, because there's so much duckweed floating on the surface. But
so what's the connection there for you? What connection are you making?
The nitrogen and phosphorus, the plants enjoy that, and there's an excess of duckweed here, so.
Cool. Awesome. Well, we'll see what we come up with, and I'll share my data with you. Who are you, though? You have a vested interest in this as well, outside of just walking up on this experiment.
So I work for Salt Lake City Trails and Natural Lands with the city, and we've been working here all summer, mainly working with the upland plants, but we're trying to plant more riparian plants down here along the stream and the lake right here.
And what are some of those riparian plants?
There's some cottonwood right there. We have all these willows along the bank here. We've got some kind of upland and riparian plants down here, a little bit more dogwoods. We've got rabbit brush, sagebrush, all that stuff.
Sounds
all
good.
The basics.
Thanks for teaching us. Do you have any advice for the youth?
For the youth? Yeah, get outside, it's great. Plantings are super fun. We're going to need a lot of people working on the environment in the future, so if that's something you're interested in, definitely follow through.
Sounds good. Thanks so much. Okay, so for our biological survey, I'm going to add muskrat. Okay, so now taking a look at all the data we collected, we know that today was cloudy. It's starting to pick up a little bit windier now, so I wonder even if that would cause a different my guess is it would cause different dissolved oxygen. Sometimes when there's more wind, more oxygen gets incorporated into the river. But as it was, we found that the water temperature was a little bit warmer in the pond. The pH, we have that moved around, was about the same. We had more phosphate in the pond here at 1 than we did in the river. We had more nitrate in the river than the pond, which is kind of interesting. We had more dissolved oxygen in the river than the pond. And I would say from that conclusion, there is, okay, so using Mitch's hypothesis that there might be more phosphorus and more nitrate, I actually want to go back and test this nitrate. Because here I'm actually questioning the sampling method, which is totally part of science. When you get some data, you can go back and retest and make sure it's reliable. And I want to make sure that that nitrate level is right. Because I would agree, I would think that maybe the pond would have more phosphate, more nitrate, there's no moving water, and because of that, more things grow, like that duckweed, which is taking over the whole pond. And because of that, there would be less oxygen due to more organisms living in the water and using up more of the oxygen as they do respiration. Both of the samples you could see through, so the refractometer had them at 0%. Let's make sure we don't have any naked numbers here. But this was 0% saturation. All right. Okay, awesome. Well, we learned so much today. We measured 2 different bodies of water, a river and a pond. We learned how to measure for nitrogen, phosphorus, dissolved oxygen, temperature. Come out with us and do it yourself. Get your hands messy, learn something new, get off the screens, and join us out here. We'll be out here tomorrow, but check the site for any other activities that we do, and I hope that you'll join us to learn more about water.